Okay, so welcome everyone to the Population Dynamics virtual seminar this is the new series of 2025. Today we have Silvia De Monte as a speaker, and I would like to share my screen to present. I think I have to share the whole screen probably, because I cannot access anymore the full screen view. So Silvia comes from the École Normale Supérieure in Paris, and the title of the talk will be Microscopic Heterogeneity and Statistical Regularities in the Dynamics of Complex Communities. Couple of words regarding Silvia De Monte. So she got a PhD at the Technical University of Denmark, and then she stayed a while in Paris, first of all as a postdoc at the École Normale Supérieure, and then at the same place as a lecturer. And now she is a researcher at the CNRS in Paris, associated with the Max Planck Institute for Evolutionary Biology in Cologne in Germany. And she's interested in ecological processes that drive evolution in heterogeneous cellular assemblies. For example, studying bottom-up and top-down the interaction of collective function and the cellular processes. How should you envision both with mathematical modeling and experimental observation, so matching the two. And some of the topic that she's interested into are self-organization in aggregative multicellularity, phenotypic dynamics of bacterial population, and evolutionary dynamics and economics. So with this one, I would like to give possibility to Silvia to share the screen. As usual, if people would like to interrupt, they are free to do it, and you can either open the microphone or write in the chat. I will read it. And so I actually suggest people to interrupt to have a better discussion. And the talk is being recorded, so if you don't want to be recorded, just click no, but you will be kicked out of the meeting. I'm sorry. So Silvia, stage is yours, please.
Thank you very much. Thank you so much for inviting me. I will present you the work of a vast set of collaborators. I would like to stress that since they are so clever, all these collaborators, this is issued by natural intelligence and not AI. Nonetheless, I have a very long title that is Microscopic Heterogeneity and Statistical Regularities in the Dynamics of Complex Communities. So thank you very much for introducing my research topics. I didn't put a slide on that, and I'll go directly to why I am interested specifically in microbial communities. Microbial communities harbor, in general, an enormous genetic diversity that has been unveiled in recent years by molecular methods. And they also have the advantage that they can be, to a certain extent, maintained in the lab, so that some predictions of models can be tested. And whereas in the wild, they are the object of extensive exploration by high-throughput genomic techniques. As you probably all know, they are essential for health, for ecosystem services like the global geochemical cycles, and for maintaining in general biotic homeostasis. What is really makes these communities different from many other communities is they are composed by an enormous amount of individuals. So it is estimated that between 10 to the 24 and 20 to 29 cells, different cells, should be part of communities on Earth. And within this plethora of cells, one can usually recognize a certain number, and here we are talking about millions of different species or genetic clusters. All these different clusters, they are sometimes aggregated into functional groups, and these functional groups have every kind of different ecological interactions. So it seems to be a system whose complexity is completely beyond the possibility of our understanding. At the same time, it seems that there are some regularities in all of this. And so people, especially coming from macroecology, have been asking if there are general features of these ecosystems that can be explored. And if there are these features, and of course, you have already understood that there are some of these features, what ecological processes matter in establishing this kind of collective or community level functions. I have been particularly interested in marine plankton. Marine plankton represents an important subset of communities on Earth, of microbial communities, despite it only represents 1% of the photosynthetic biomass, which is mostly composed by trees. Nonetheless, it is responsible for nearly 50% of the Earth's primary production and constitutes 85% of the pelagic biomass. I delved into this magical world of plankton, in particular by collaborating with the Tara Oceans expedition, that is the most extensive study of planktonic communities that has used exactly the same tools and protocols in many different sampling stations. And here you have just a representation of the Tara Ocean and part of the Polar expedition, but there are also other missions realized in the Pacific. Actually, no, there's also the Tara Pacific. So there's a really huge data set that is available. And in the first analysis that have been realized on this data set, people, of course, started to count how many different species were there. So just for those who are not familiar, species, the concept of species is a bit tricky for microbes. So people started to count, first of all, the number of different genetic clusters that were detectable in this data set. And it turns out that putting together all the data from, in this case, protists, so unicellular eukaryotes, they came out with an estimate of about 100,000 different species. This dataset has also a very nice feature that these measures are realized at different levels of organism size. And so when people compared the diversity computed as a Shannon index across different organism sizes, they found that whatever is the size, this diversity is very high. But what was really surprising to me is that despite this a huge variation in the environment where these samples have been taken, the amount of variation in diversity was relatively small. And this was something that was quite unexpected, because one would have imagined that in, planktonic deserts or oceanic deserts, perhaps diversity would be different as it is in deserts on land. In reality, this existence of a very huge amount of diversity had been already observed and is something that is really characteristic of microbes. This existence of a huge number of rare taxa is usually known as the rare biosphere or the microbial dark matter. And one can see this in this kind of representation where you rank the abundances of species from the most abundant to the least abundant. And then by looking at these distributions, you can differentiate between some cases that are typically of high diversity, where the tail of this distribution is pretty fat, and other cases where the abundances decay very fast. So the kind of questions that we started asking on by analyzing the Tara Oceans dataset is, does the rare biosphere in this dataset obey some specific laws? And if it does, what are the processes that shape it? And then we went on asking what kind of ecological interactions matter for establishing these laws. And in particular, we wanted to understand whether differences between species or between habitats, between locations, were actually important or not in quantitatively in determining how rare species are distributed.
Just a second, we have probably a
Yeah.
question from the audience. Would you like to speak out, Preval?
Yes. Hi,
Yep.
so Silvia, I had a question.
Yeah.
In the previous slide, when you talk about rare biosphere, there seems to be a cutoff. Is that arbitrary, or is there something determined that zero 0.1 relative abundance cutoff for rare biosphere?
No, this is, yeah, I'll get to that.
Okay.
So this is, usually it is set arbitrarily.
All right. Yeah,
But
thanks.
I agree, I don't like this arbitrary thresholds. So I'll explain to you what we did, which was exactly in order to avoid to set arbitrarily a threshold and decide what is rare and what is not.
All right.
So what we did was that we took the samples, protist samples from the Tara datasets. So we had nearly 400 samples that were coming from more than 100 locations in the world ocean. And again, 4 size classes. And when we plotted the rank abundance distributions, we realized that despite some variation in the part of the distribution that is relative to very abundant species, the tail of the distribution that is here represented in a double logarithmic plot seemed to be very similar across all the samples. So we wanted to make this more quantitative, and in order to do this, we devised an adaptive algorithm that was actually fitting this distribution starting from the tail and keep going until a certain tolerance that we would set so that the fit of the species abundance distribution was believed to be satisfactory. Here you have just one example for the data that are displayed here with the filled dots. So the problem with this kind of exercise that had been done not with an adaptive fit, but had been done previously by fitting all the data, is that you need to choose a law that you want to fit to the data. And there are many different possible functional choices that you can take. So the choice we did with Enrico was to take one model that is a semi-neutral model. I will not go into the details, but we can discuss it later if you want. That provides a distribution that is a combination of a power law and of an exponential decay, so that the 2 main classes of decays were represented in this functional form. And by doing this, we could fit the 3 parameters that define this distribution for every sample of this dataset. And by doing this, we could also compute the dominant, the trend of the power law that is the typical decay for this dataset as a function of these parameters. So the lambda is the exponent of the power law decay. And by doing this, we realize that this power law decay is vastly the same everywhere in the ocean. So we could not find any correlation that was stably maintained with any of the metadata that were associated to this dataset. So with environmental conditions, with biotic or abiotic factors. And it turns out that the exponent has such a small variation, so putting all the data together, the coefficient of variation is less than 10%, that it seemed to indicate that there is some kind of universality or, I mean, something behind this. This exponent is actually telling us that the decay of abundances of rare species, and by rare species I mean 98% of the OTUs that were present in the dataset. So this decay is the same everywhere. To extend, so a recent publication has extended this observation that what they call the power band distribution, that is again the product of a power law and of an exponential distribution, seems to be the best one to describe a huge data set of abundance distributions. So you see here we are talking about thousands of different data sets, different samples. In this case, what was tested was a role that has sampling in distinguishing between this shape and the Poisson log normal shape, that is the shape that is most commonly used for explaining environmental data sets. Unfortunately, in this work, they didn't really look at the value of the exponent, and results are a bit all mixed together. But this also indicated that there seems to be a very strong prevalence of a power law decay with an exponential cutoff. However, there was something that we were not very satisfied with in terms of the model we used for fitting our data. And this was that it was coming from a neutral or semi-neutral model that did not include interactions among different OTUs or species. And this is something that seems very unrealistic in the ocean, where all species are sharing a same relatively well-mixed environment, and they are obviously competing for resources. They have blooms, booms and busts that can be seen from, as you have seen previously, from the satellite. So we started together with Emil Malmin, who is a postdoc in Plön, and Arne Trausen, to analyze a different kind of model that was including some more realistic, in our view, ecological hypothesis. The model we chose is basically the simplest nonlinear model that includes interaction between different species. That is the generalized Lotka-Volterra equations. In these equations that are purely deterministic, one accounts for the variation in time of the abundance of species I in terms of intraspecific competition, interspecific competition, and an immigration term that we added in order to avoid species to get permanently excluded, so we didn't have to include also, evolution, mutation, and so on and so forth. This, I'll go back to this immigration term, which is quite important actually. So we have communities that are composed by a number, big S, of a large number, big S of species, and we choose pairwise interactions so that in a way that is a bit different from what is usually done, for instance, in statistical physics, where these equations have been extensively studied. So we chose to fix the average and the variance of this interaction matrix independently of the community size, which means that if you add another species, then every other species will experience more competition on average. And then, by simplicity, we took correlations equal to 0. So this, otherwise, this matrix is, the elements of this matrix are randomly chosen once the statistics are assigned. So the interaction matrix would look something like this. Now, since interactions are strong and S is large, then what is expected is that species will experience a large amount of competition, and therefore the principle of competitive exclusion should apply, or how it is known in ocean science, the paradox of the plankton. That is, one would expect that just a few species will come, one or a few species will come to dominate the whole ecosystem. So when we look at simulations of those equations, we see that at any given time, indeed, the community is mostly composed by a small number of dominant species, and all the rare species you can't even see. So here you have 500 different species, but you don't really see most of them. However, when you let the time go, you realize that this dominance by a small subset of species is ephemeral. And the clique that is dominating at a given time is subsequently superseded by another clique of a small number of species. And this turnover goes on forever. We try to understand what did this mean in terms of community composition, and it turns out that the system is exploring the vast phase space by approaching progressively some low-dimensional equilibria that can be identified by throwing away at a set time all the species that are rare. You only keep the dominant ones, and then you let the system go to equilibrium. If you compute the distance between the real trajectory and this low-dimensional equilibria, you will see that so the similarity between these two, that is the inverse of the distance, actually goes to 100%, very close to 100%. So this equilibria are approached, and then the system goes away. So at any time, this community looks like it is effectively low-dimensional. However, the high dimensionality of the system is necessary for this turnover of guilds to be maintained. If the system is too low-dimensional, you would only observe that the system goes to equilibrium. So this idea that there might be all the time a turnover of species between a rare component of the community and the dominant component of the community is somehow reflective of the seed bank hypothesis. However, the difference is that we do not need here to suppose that there is a dormancy stage or some additional ecological process other than competition with the other species. So what I showed you is just one example, but it turns out that this kind of high-dimensional chaotic trajectories are observed in a vast region of the parameter space, where the parameters are the mean and the standard deviation of the interaction matrix. And I remind you, the interaction matrix here has no structure, is randomly chosen, and this is why you see that you don't always have the same outcome because the realizations of the matrix differ from one simulation to the other. So on top of this chaotic region, you also have other phases that have been extensively studied. So when competition is too strong, you have competitive exclusion. When competition is very weak, then you have a unique fixed point equilibrium where many species coexist. And this region of multiple attractors is very complicated. There are some people who are now looking into it, but let's say it's a region with multi-stability. If one looks more in detail to exactly what happens in this system, one can realize some adiabatic simulations where you are actually maintaining the same structure of interactions and changing slightly, little by little, for instance, the mean or the standard deviation of the interactions. And then you can see that there are some kind of radial structures that emerge that can be actually explained as I will perhaps mention later. So still, this is the chaotic region, so consistently chaotic. What is interesting of these chaotic attractors is that even though this whole region is chaotic, the number of species that are dominant in the community is not the same for all the parameters. So if one measures the effective dimension of the community as the inverse Simpson index, that is a typical metric in ecology, one finds that in this region parameter space, the dominant community is smaller, and it increases, actually, in principle, it diverges as the system goes towards the fixed point equilibrium. So this idea that the number of effective degrees of freedom, or if you want, of effective species that are really dominant in the community could be perhaps detectable in data, led us to collaborate with Steve Munch at the University of California, Santa Cruz, with whom we are now testing whether his methods of Bayesian inference of the maximum Lyapunov exponent from time series can be applied to this specific model, to the simulations. And if this is true, then we would like to apply it also to natural time series. Always Steve and colleagues, in particular Tanya Rogers, found that actually chaotic dynamics is not as uncommon as previously thought. They reanalyzed a vast number of time series and found that in particular for phytoplankton, the vast majority of time series held the signature of chaotic behavior. If you want to see more or less how this looks like, this is a very time-resolved time series obtained. So these are OTUs from barcoding of a bacterial community. And you can see that in this community, OTUs have a very intermittent kind of variation. Many of them remain rare for most of the time, and some of them have this boom and bust dynamics. And this boom and bust occurs on a time scale that is much shorter than the seasonal variation. If now we go back to the model and we look at one species at a time, here you had the stack plot, and now we look at the single species, but for a much, much longer integration time, we find that every species in this community has its moment of glory. So there is no species that is permanently excluded from being dominant. But as I will discuss later, there are differences between different species, but in general, the dynamics is always this intermittent boom and bust. The signature of this kind of dynamics can be seen in the abundance distributions. So let me explain this figure that contains a lot of information. So you can look at the community at a given point in time. And then what you would obtain is what is called the species abundance distribution. That is, this is represented by this histogram in blue. So here you will have that different species have different abundances and contribute to this distribution. Alternatively, you could less in the data, but more in the simulations, you can just look at one single species and then look at what is the distribution of its frequency over time. And then with the very, very long integration, you will get another distribution. And now what you can see here is that these distributions are basically overlapping. So the black line is the average of all the distributions of the frequencies in time of all the different species, which means that to a certain extent, all species are equivalent in the way they behave, even though each species in this model has different interaction coefficients. So we wanted to understand a bit more why there is this sort of emergent neutrality in this model. And to do this, Emil came up with a very nice focal species model that reduces the complexity, massively the complexity of this system. So this model is representing the typical behavior of any of the species in the community by means of this very simple logistic model where the growth rate is fluctuating in time according to an Ornstein-Umbeck process. So this is colored noise. And by using this model that can be analytically solved in some limit cases, one can get a distribution that looks very much like the one that is simulated. Okay, the dashed line is the analytical approximation, and as you see, it doesn't behave all that bad. So this model is basically capturing the effective neutral behavior of species in these communities. And its approximation, this color noise approximation, allows us to estimate what is the value of the power law exponent. And now if we compare this value to the value that is measured on the simulations, we find that this provides in general a very good approximation. You can see here that the value of this parameter is basically between 1 and 1.5. So it's a bit lower than the value that we see in the data. Perhaps, well, I'll skip this, but it's just to say that there is a way to compute the parameters in this equation from the time series. So this is applicable in principle to data. So we can also see that species are different from one another on top of this neutral behavior that is shared by every species. Indeed, we can rank species between those that whose bursts and busts as well are occur more often to those that have their bloom, let's say, less often. And this ordering is connected to something that is not actually scaling with the number of species, because there are some intrinsic differences in between one species and the other with respect to the average species in the community. So to conclude, and I put here this nice video that was realized because this project led me to interact with some artists and in a very nice art science experience. Anyway, so strong competitive interactions in disordered lot-Cavalieri equations drive exclusion on a short time scale. But in the presence of weak immigration that avoids species to go permanently extinct, the community composition keeps turning over. in a way that there is really no winner in this community and no permanent loser. The effective dynamics of an average species in the community is well approximated by a focal species model that captures basically the neutral nature of this system, neutral in a broad sense, but that is grounded in the hypothesis that the community does not have really a structure here. So there is a symmetry between species, not in the sense that are identical, but in the sense that every species has parameters that are drawn from the same distribution. So our next question was, what happens if there is some structure in the community? And we know that microbial communities are structured. So this is a nice study in the lab where people have shown that actually when you clump communities in functional groups, you get much more understandable regular behavior and reproducible behaviors. However, interactions are not only due to what functional group species belong to, but there is also a number of microscopic details that differ from species to species. So we wanted to try and complexify a bit the model with respect to the classical Lotka-Volterra equations studied in disordered systems by adding to this disordered part also a structure that comes from some, let's say, previous ecological knowledge of the community. And this is work mainly realized by Juan Giral Martinez, a PhD student that I co-direct with Mathieu Barbier. So now we have a different kind of scaling here. So this random component is actually scaling with S to keep with the usual assumption in statistical physics. And now I'm going to tell you a bit how we define this structured matrix. So the types of structure that we were thinking we would like to represent are, for instance, group structures. In this structure, you have that all species that belong to the same functional group are supposed to interact in the same way with all species that belong to a different functional group. Or you might have an S structure, for instance, something that comes out when interactions are driven by traits that have evolved, and here you have a signature of the phylogenetic tree. Another kind of structure that is very much studied is a structure that derives from the existence of common resources, and the competition for these common resources or the contribution to common public goods will also provide a structure. So Juan thought, okay, we can describe all these different cases by considering that on the one hand, we have a set of species, and on the other hand, we have a set of functions at the community level. So these functions could be the functional groups, or they could be, for instance, resources. And now each species will have an effect on some of these functions, and in return, functions will have an effect on species. So every species will be actually defined by two traits, the effect traits and the response traits. And just to give you concrete examples, let's say I have two functional groups. Now all the species that belong to functional group 1 sum up in their abundance in order to determine the total abundance of species of functional group 1. And this total abundance will influence species of functional group 2 depending on these response traits. So the contribution, the effect rate will be the same for the same equal to 1 for every species of group 1, whereas the response traits will be in general different for the species of group 2. A different case is a consumer resource model. So now every species can contribute, perhaps consuming, perhaps producing, to some of the resources or public goods, and then in return, every species will have its own growth rate influenced by this community-level degrees of freedom. Now, with this kind of formalism, starting from these two vectors that are the effect and I forgot the name. I always forget. And the response rates of every species that are here represented in the case, easy case of three functional groups. You can derive the interaction matrix in the following way. So you will have in general an interaction matrix that is of low rank. And once you have this matrix, you can use dynamical mean field theory, that is a technique of statistical physics, to derive an equation that is describing the abundance of species I in terms of its own abundance. This is the usual, self-regulation term. A term that represents the fluctuations due to the rest of the community, and a term that is usually not present in the MFT, or at least is simpler than this one, that depends on the existence of these collective degrees of freedom. Which means that you have reduced the complexity of the whole system to just nf+1 equations for the mesoscopic degrees of freedom that correspond to the functional the functions of the community. So NF is the number of functions of the community. And these degrees of freedom are actually related by self-consistent relations. So to some extent, you can forget, this is telling you that you can forget the microscopic details and just focus on the functions. So let me explain what this means in practical terms. So suppose you are interested in looking at the species abundance distribution for a community that has some structure. So perhaps you will have 3 or 4 different functional groups. So the previous equations are telling you that at equilibrium, you will find that each functional group will have a certain distribution of abundances, but this distribution of abundances will overlap altogether at the community level. This also tells you that if you want to obtain, for instance, a power law distribution at the community level, you need somehow to have that the functional traits are distributed by a power law, which is something that was commonly assumed in models and has some, it makes some sense if you think of allometric relationships for microbial communities. So now, just in the last 5 minutes, I'd like to say briefly something that is a bit more complicated, but I find very exciting. That is what happens when the system is out of equilibrium. So out of equilibrium dynamics is very often found in models with very few degrees of freedom that describe ecosystems. So for instance, phytoplankton, zooplankton, predators, detritus, or functional classes. So what happens now if you think that within each of these functional classes, you don't have just one species, but you have many species that are different from one another? And you want to introduce this heterogeneity in the way we have done previously. So let's consider a very simple model where you have 3 functional classes that are in a rock, scissors, paper relationship. That means that when all species within a functional group are the same, you will have that they will all oscillations, and these oscillations will be the same for the functional group and for every species that belong to them. And now you start increasing the spread, the heterogeneity between each of these functional groups. What happens is that these oscillations at the species level become more complicated, they dephase, and at a certain point, the system loses the capacity of oscillating. And if you keep increasing heterogeneity, you will get into a phase where all the species oscillate chaotically, but their average remains constant. So to draw this in a phase diagram, what I have illustrated here is a progressive improvement of microscopic diversity that leads to two kinds of transitions. One transition is already well known, and one can call it the May transition. So it's a collective transition from an equilibrium to a case where there are asynchronous oscillations. So from the point of view of the community, this is not so much different from the macroscopic viewpoint because everything is at equilibrium, is averaged out. What is interesting is that there is a low-dimensional bifurcation that leads from an out of equilibrium dynamics to equilibrium. So which means that diversity is stabilizing and not destabilizing the ecosystem, that is contrary to what May had predicted. So this happens also when you take more complex out of equilibrium dynamics, for instance, chaotic dynamics of a low-dimensional system. You will have more complicated bifurcations. So if you start from this chaotic regime, you will get all the way to stabilization through an inverse Feigenbaum cascade. And what is surprising is that if you take my first model, where you have a huge amount of species and where chaos is due to the interplay between the species, and now you say, okay, each species might have different strains, and now I am increasing the heterogeneity of these strains, you obtain exactly the same kind of scenario. So as you increase diversity, you are actually stabilizing the system before you go to the asynchronous oscillation stage. Okay, this is actually, I didn't have time to go into the details, but these features can be understood by looking at the Jacobian of this class of systems. So if you are interested, I let you have a look at this preference. But in general, the conclusion is that when you combine structure and randomness, you can actually complexify or not the conclusions of simple systems depending on the amount of randomness and amount of heterogeneity. And now the big question for us is how can we evaluate this degree of randomness in real ecosystems? And before I finish, I would like to advertise this very nice school that we are organizing for next September with some wonderful speakers. And I want to remind to all young PhD students, postdocs, or young PIs to apply before February 28. And thank you very much for your attention.
Thank you, Silvia, for very, very nice talk. I will